Hello, everyone. Welcome to LakePharma webinar, part 2 on reagents and assays for COVID-19 antibody response. My name is Walter Tian, VP Commercial Operations at LakePharma, and I will be the moderator for today. For those of you who attended our part 1 of the webinar, which was about a month ago, thank you and welcome back. And if you missed the first webinar, I do want to let you know the recorded version is available for viewing on our website. So make sure you take the time to check it out. Before we start the webinar, a couple of items. First, all the attendees are on mute. So you can submit questions through the Q&A box, which is on the toolbar to the bottom of your screen. You can do that throughout the webinar. Now, let me introduce the speaker today, Dr. Hua Tu. Hua is the founder and CEO of LakePharma. Hua started his biotech career in the Bay Area, started with Telluric, later on part of Amgen. Hua started LakePharma back in 2009 in a single lab space in Belmont here in the Bay Area. Over the last 10 years, built LakePharma into a comprehensive integrated solution providers with facilities in 3 states, 6 facilities. So today, Hua will focus his webinar content on the new development on COVID-19 reagents. So with that, I'll turn this over to Hua.
Okay, thanks, Walter. Hi, everybody. Thanks for joining us today. This is a follow-up to the webinar we did about a month ago. That would be part 1. Both part 1 and part 2 are focused on the reagents and assays related to the COVID-19 antibody response, so against the SARS-CoV-2 virus. If we can go to the next slide, please. Just as a brief introduction, LakePharma is a US-based biologics CRDMO with operations in California, Massachusetts, and now in Texas. The company provides integrated solutions on research, development, and GMP manufacturing of biologics. And since January, we have been active in 3 areas against COVID-19. We started with protein vaccine program and generated a range of SARS-CoV-2 proteins. And then we realized those proteins are important and useful in the serological testing. So then we start to apply those proteins in the assay side. And the third domain we work on is the for therapeutic antibodies, which is a traditional business, standard business at LakePharma, where we can synthesize and produce large number of recombinant antibodies and do a range of testing. Walter, if you can advance 1, please. So all 3 activities, even though they have the different domains, but they actually require similar level of a similar level of research. So, and they're common in that they all need the high quality antigen, require antibody binding assay, neutralization assay, as well as understanding the mechanism. So that's the topic for today. Next slide, please. This slide number 6 is a recap of the part 1 webinar we presented about a month ago. We showed the characterization data of various COVID-2 spike proteins and RBD, and as well as the ACE2 binding assay. And we also have made the reagents available to the community, primarily with COVID-2 and RBD as antigens for serological assays, also known as the antibody assays. We make the product economically appealing to the community. So we price at $450 per milligram for the full-length ectodomain spike protein trimeric form for nonprofit, and the same protein, if it's used for commercial diagnostic companies, we charge $900. So $450 per milligram is about our production cost. Okay, this is not a. We didn't enter into this business to really look at it from a revenue perspective. Our motivation is to produce high quality proteins and make them available broadly to the community, so everyone would be looking at using the same reagents for much better comparability analysis. So far, our proteins have been sent to at least 25 organizations, and in the early days, we did ask for a data bag, and we want to get feedback on the results. And I'm happy to report that the results are satisfactory in the serological assays. Now, the part 1 webinar is captured and posted on the LakePharma YouTube channel, and you can view that over there. Next slide, please. The specific topics for this presentation is we want to show new data we generated with the COVID-2 spike protein, and we also want to discuss the reagent availability with regard to spike proteins from other coronaviruses. We have generated those. Then we'll talk about the virus neutralization assays in various formats, and we want to save some time for the Q&A section. Next slide, please. So the spike protein, again, we committed ourselves to be a source for medical centers and nonprofits, and we make the reagents at $450 per milligram. And I think we actually got quite some feedback on the price point. I think people understand that our price we price it very, very low, and that's well, again, that's our intent. We want to make sure the proteins are available to everybody. More important than that is we also want to make sure we can deliver the products in time, so that so as a customer, you're not waiting to get your proteins from us. So, and at this point, we pretty much have caught up with the on the supply side. We are in gram scale production already. We are scheduled to produce gram scale weekly, so there's about at least a gram produced every week. We continue to optimize the manufacturing process. We are at a process that is robust enough for us to ensure the gram scale production. Spent quite a bit of time establishing release assays for in vitro diagnostic applications, and this is where we have had discussions with a number of institutions and diagnostic companies. What we have come up with is greater than 90% purity by SDS page, and then greater than 80% trimer form by SEC. We actually, with the spike protein, we have not seen the presence of the monomer, so the molecule is primarily trimer. So that's those are not too difficult to achieve. As a third criteria for the release assays, we want to ensure a product actually bind to have functional validation, and we established that as the binding to recombinant ACE2 Fc protein, and we have a reference standard of the spike protein already established. And we want to ensure that the binding profile is consistent over time. There's quite a bit of formulation and stability study have been done on this, and we have a desired product stability profile of stable at 4 degrees for at least 14 days, and make sure the protein can be frozen, and storage stability at -80 for a good period of time. Our freeze and thaw studies, and we have come up with a formulation that allow freeze and thaw, and we see that freeze and thaw does not significantly impact the ACE2 binding. We also have done stress test at 40 degrees for 3 days, and it appears that protein actually handled that condition pretty well, and it does not significantly impact ACE2 binding. Next slide, please. Now, the ACE2 binding curve is showing on the right, and it's an assay that we run routinely now. Next slide, please. One more click. Okay. The past few weeks, we actually have done quite a bit of looking at stability, and we knew that the spike protein is well, it's a hard protein to make, and we noted the potential stability issue a month ago, and now we have some data, and we found that the purified spike protein is actually sensitive to agitation. And if you agitate it, that can lead to particle formation as well as precipitation. We also have tried to achieve a higher concentration with the spike protein, but we found that the protein does not appear to like 10 milligram per mil kind of concentration, as that can result in precipitation as well. We have now sent the protein out for cryo-EM analysis to understand conformational changes. So there will be more data on that in the future. We don't have that data now. With the data all together, our current thinking is that we have a way to keep the protein stable at 4 degrees as well as frozen. The product concentration is now set at 1 milligram per mil. We can go a little bit higher, but we're shying away from a high concentration formulation such as 10 milligram per mil. For most ELISA applications, even 0.5 milligram per mil will do, because the protein product will actually be diluted massively before applying to the plate. So now it's the new production is all going to be at 1 milligram per mil. We'll continue to ship the 1 milligram. So the 1 milligram samples will be placed in the 2 ml tubes, and we'll continue to ship that on ice pack. But for larger samples, we, in order to avoid the agitation that could encounter by during transportation, any vials that are 10 milligram or more are shipped frozen with dry ice. We continue to like the tris-based buffer, and we think that the most stable, it appears to be more stable than PBS. But for certain applications, such as conjugation, a tris-based buffer would be less than ideal. And for folks who still want the product to be in PBS, that can be done, because after purification, we have a buffer exchange step anyway, so we can buffer exchange to PBS, but we would only do that for larger orders. And maybe custom orders. I see there's a question asking, is lyophilization an option? We believe so, although we have not done it, and to my knowledge, none of our partners have done it either, because the protein can be frozen. Okay, it can be frozen. Therefore, we do think the protein should do fine with the lyophilization, but at this point, we don't have the data yet. We may get to do that fairly soon, and we'll share the data. All right, I'll save, I see a few questions already. I'll save some of the questions later. Next slide, please. Okay, yeah, the slide 10 on glycosylation. We have done mass spec-based glycosylation studies with the spike protein and RBD protein we produced, and we definitively can see that RBD contains 1 O-glycan and 2 N-glycan. The spike protein, the glycosylation is a bit more complex, and we actually do not have the entire picture on that yet. So nevertheless, we can appreciate that the N-glycan on the spike protein forms mostly high mannose and complex glycans. So this is something that it's actually pretty challenging to really get an entire picture. Advance, please. We're happy to see there's a recent paper published, and this is what's shown here in this ring circle glycosylation profile. That's not our data. That's actually taken from the publication by, I believe it's Georgia, and where they have analyzed the glycosylation of the spike protein they produced in, I believe it's in the mammalian cell culture. And the data they show overall, our glycosylation data agree. Our data may not be completely consistent, but they also pick up the O-glycan on the RBD. So there's going to be some more work to be done there, but it's right now I think we have enough of a basic understanding of the glycosylation, so it's not a very active domain for us in-house right now. Next slide, please. We have also established now the ELISA assay in a serological assay format, looking at human plasma samples. We were fortunate in that we're able to secure a pretty good amount of human plasma donation. And we have tested those plasmas. We found some of them actually have good titers. We have more samples with no titer. So nevertheless, they form a panel that we can use to test product quality and consistency in a serological setting. And the graphs, the titer serodilution graphs in the ELISA is shown below. We continue to use ACE2 protein as well as the MD10 antibody as reference standard in the ELISA assay, but now we have a few human plasma samples that are high in titer of the antibody. So that's something we start to use and as a way to test the product quality. That's the product quality of the spike protein, and as well as understanding the consistency in the ELISA assay. In addition, we have also now started to generate or receive recombinant antibodies, and we're using those antibodies in antibody binding assays. We're actually seeing a wave of antibodies coming in, and there's a range of antibodies that have been established, and it's not just antibodies bind to RBD, but antibody binds to really all other parts of the spike protein. So we intend to build a panel of antibodies as references. So I think that may be a reagent quite useful, and we perhaps will cover that in a future webinar. Next slide, please. So I want to move on to the second topic, which is looking at the spike protein from other coronaviruses. Well, we believe the spike protein from other coronaviruses are important references for serological testing, as well as for antibody testing. And other coronaviruses are already in circulation. And here, I would send a paper by folks in the DoD, and it's a 2018 paper. It's worth reading. Of all influenzas, this paper showed that 12% of subjects with influenzas are PCR positive one way or another for a coronavirus. And the popular ones are OC43, 229E, NL63, as well as HKU1. And the percentage number in parentheses are the abundance, the distribution of it. So those are 4 that are definitively in circulation already. So we are, in addition to that, we also made the MERS and SARS spike protein. We made all the protein the same way, which is full-length ectodomain, trimerform, and in tuna CHO. So what I show you here are the data for the spike protein from SARS, MERS, OC43, and HKU1, and that's our initial reference panel. We tested the protein in a range of assays, in addition to SDS page, SE, and also for the ACE2 binding assay. Out of the 4 molecules shown here, really only SARS can bind to ACE2. MERS does not, OC43 does not, and HKU1 does not. And that is actually consistent with the literature. SARS also used ACE2 as the receptor, whereas MERS, we believe they use the DPP4 as the receptor. So those are reagents that are now available in LakePharma. Next slide, please. Moving on, I want to spend some time on the neutralization assays. There are 3 kinds of neutralization assays that are being run in many parts of the world. Okay, I think that the most robust and most relevant is the live virus infection assay, and that's using real virus, live virus of SARS-CoV-2, and because it's using the actual virus, I think it's the most physiologically relevant. The challenge of doing that experiment is that you will need a BSL-3 facility to do that. And that's not an easy task, and that's not going to be broadly available. The second level test is something that's very popular in the virology space for many years. That's called pseudovirus infection assay. And this is to make pseudovirus with the spike protein expressed at the surface using recombinant DNA technology. And those viruses can infect cells, but they are incapable of replicating. And most of them use the lentivirus platform for that. So, and the infection is reported using a reporter gene assay because the pseudovirus will contain either a GFP or luciferase or something that would trigger a GFP or luciferase response. So it turns into a reporter gene assay. This kind of assays are very popular, and you would not need a BSL-3 to do that. A BSL-2 would be sufficient. So it's popular, and we also believe that it's quite relevant because it is done in a control way, and you can actually test a range of viruses, multiple pseudoviruses together side by side to look at the response. So it's a much better controlled experiment. There's a third kind of assay that is not virus at all, but looking at cell-free assay for looking at the interaction between ACE2 and spike or RBD. So the advantage here is that it's very much focused on interaction between spike protein and the receptor. It's very mechanistic. We know exactly what that means is the disruption of the interaction between the two. But the disadvantage here is that it's limited to one mode. So you could have an antibody, for example, that can block the virus entry, but it's not, the mechanism is not based on the inhibition of the RBD and ACE2 interaction, then you would not be able to capture that in this format. So that's the issue here. But the advantage on it is that it's a relatively easy assay to set up, and there are various format that can be used. For example, you can put ACE2 on plate and have the spike or RBD in solution and look at interactions that way. And you can do it reciprocally by putting the RBD or the spike protein on plate and have ACE2 in solution. They all can be done. In fact, I think we'll probably have done all of them already. What we have chosen is what we have focused on is the first format, which is have ACE2 on plate and have spike protein in solution. Next slide, please. Next slide. And I want to show you some of the data, and there's some surprising data here. Let's focus on the left panel first. In this experiment, we have the ACE2 on plate, and we put the full-length spike protein in solution. And we added a range of reagents and look for the ability to inhibit, to block this interaction. Okay. And what we see is that ACE2 can block this interaction. That makes a lot of sense. And we chose three antibodies here. There's MD10, which binds to the his tag. It doesn't block the interaction. And sure enough, it doesn't block. We also applied two antibody. One is a recombinant IgG, and the other one is the recombinant IgM. This IgG and IgM actually, they have the exact same variable region. We just made those as a control. And this particular antibody, it binds to spike protein. We know that, but it doesn't block this interaction. And in this case, it doesn't show any inhibition. So we think the assay is actually it's pretty robust in this case. But while we're doing the experiment, we noticed something strange. Again, focus on the left panel, the IgG and IgM here, they didn't bring down the signal. Okay, we understand that because they're not neutralizing antibody, but they actually increased the signal. And that was a little bit bizarre. So we actually look into that further, and now we look at the panel on the right. We still have the ACE2 Fc on plate. Now we have RBD in solution. Now we're seeing a whopping effect here where the IgG and IgM and even MD10, instead of keeping the signal the same, it actually increased the signal dramatically. In fact, so dramatic, we had to stop the reaction where you don't see much of you don't see a whole lot of ACE2 inhibition here because the plate is already developed very, very robustly. And we actually we scratch our head for about 30 minutes looking at the data, and then I think kind of all makes sense. And the reason we're seeing an increase in signal instead of a decrease in signal is because RBD is a monomeric protein. And it has interaction with ACE2FC, but once it's bound by an IgG, which has 2 binding domain, or an IgM, which has 5 binding domain, this really established an avidity effect. Those antibodies actually now make the RBD bind to ACE2FC much more efficiently. But we think that's actually just due to avidity. Now, we like this assay. We were concerned about the two-directional aspect of the assay, but now we actually like it because it's now pretty clear in that if we use this assay, blocking antibody will decrease the signal. But if something that does not block but can bind, it would actually increase the signal. And if you have an antibody that doesn't block or doesn't bind, then it would not change the signal. Okay, so it's actually quite telling over there. So we have been using this kind of cell-free binding assay as a form of looking at neutralization activity, and I want to show you an example in the next few slides on how we're using that. So next slide, please. As I mentioned earlier, LakePharma actually has a whole team doing protein vaccine development against the SARS-CoV-2. And the vaccine candidates have been through several animal studies now. And I'm not showing all of the data. In fact, I'm probably showing 5% of the data they generated, because I want to show the data in the context of neutralization assays. So this particular data I selected for this presentation is actually is one candidate for the protein vaccine. It's not the one that's most immunogenic. I selected because it shows you a clear time course, as well as this particular immunization is done with a commercially available adjuvant. So anyway, if you see the titer is not impressive, well, this is not the one with the most impressive titer data. So we did immunization and look at the titer at day 9, day 16, and day 23, and we're seeing titer in the mouse plasma. This is the mouse experiment, and specifically, this is using the LakePharma Pentamize platform, which is developed by LakePharma's Discovery Immunology business unit. Brian Zabel, our senior director of Discovery Immunology, gave a webinar on Pentamize about 3 weeks ago. I believe that recording is also available on the LakePharma YouTube channel. And what we see here, this is the Pentamize, and the Pentamize has the uniqueness in that it's an immunization platform, but it actually has MHC strains. It's using different MHC strains in it. And what we saw here is at day 23, we're seeing titer of various kind in different animals, but we actually see good correlation between titer and the MHC strains. So with that, we took when we analyzed the neutralization activity from the plasma and used the assay I just described in the past few slides, we're seeing that neutralization activity correlate with the titer. And this is an observation we have made with multiple immunization campaigns. So we do believe that this type of neutralization assays is actually quite robust and correlate very well with the titer. So we probably will show more data about our protein vaccine the development in the webinars and communications in the future. Let's move to the next slide. We then took the same plasma we analyzed took the same plasma we analyzed for titer assay, for neutralization assay in LakePharma. Then we sent it out to Chicago, to UIC for the pseudovirus neutralization assay, and that's a virology lab who's been doing this for many years. And the data is provided by Laura Cooper and Li Junrong at UIC. We send them 5 plasma samples, and if we look at the top panel middle, where the 5 plasma, they took a 1 to 100 dilution of the plasma and applied to the SARS-CoV-2 pseudovirus assay. They saw 100% inhibition with plasma 1, 2, and 3. They didn't see much inhibition with plasma number 4 and 5, and that makes perfect sense because 1, 2, 3 are the ones with a lot of titer, and plasma 4 and 5 are our negative controls. So this shows that our plasma from the vaccine studies actually have can neutralize the pseudovirus in this assay. As reference, the plasma have also been applied to the SARS-1 pseudovirus as well as VSV. Now with VSV, we're not seeing much of a difference. It appears to be, there's a background about 20% over there. With SARS-1, we also do not see significant inhibition. There may be a 30% inhibition with plasma number 3 and 20% inhibition with plasma number 1. So that might be from some level of cross-reactivity where the antibodies we generate using SARS-CoV-2 that may cross with SARS-CoV-1 at some level. And this is not surprising because we have already seen in previous cases where there's some cross-reactivity between the two because the two proteins have some level of similarity. They then took the plasma 1, 2, and 3 and did a serial dilution, and we could actually see IC50 for plasma 1 at 1 to 2500, plasma 2 at 1 to 750, and plasma 3 at 1 to 3300. And when we took that data back and correlate with our titer as well as neutralization data, that actually make perfect sense. They correlate very, very well. With this data, we believe that the ACE2 binding ELISA in a cell-free format is a useful assay to look at potential neutralization activity. So, all right, move on to next slide, please. I'm near the end of this presentation. I want to summarize it again from a reagent availability point of view. For spike protein, we make RBDs, but majority of our work are with the trimer form, Histag, C-terminus, and we make them in the tuna-chill transient format. We continue to support the community with SARS-CoV-2, and at a price point I hope you find interesting. And I also hope you find that the quality of the product is satisfactory. And if it's not, please let us know, and we're happy to hear back. The spike protein from other coronaviruses are now also available. We do not intend to sell those proteins. So those proteins are available for antibody testing within LakePharma. Just a little advertisement for the LakePharma Antibody Center. Our group in San Carlos, California has a wide range of antibody testing capability. So you're welcome to, if you make antibodies, you can have them test. And if you generate your antibodies in LakePharma, then we probably we'll throw that in for you to test antibody. ACE2 Fc protein, I continue to believe this is a good reagent, and we have made the protein. That protein is available. We make it very, very economical. I think we charge $100 for a vial. We want you to use this protein. Now, if you're going to buy this protein in large quantity from us, we might actually have to charge you more because we have to go into custom production in order to make this protein. So we have made recombinant IgG and IgM, and I showed you the data earlier. Those proteins, again, they're not for sale. They're available to you as a collaboration, and they're available to you for your antibody testing in LakePharma. but there's no price point on those. And the last one, well, that's not our product. It's a product we use a lot. That is the recombinant anti-His antibody. And it's MT10. It's a human IgG backbone. It's highly specific. That product is available from Mena Scientific. Next slide, please. This is my last slide, and again, this is on the resource. We do put information on LakePharma.com, and we have specific pages on the COVID-19 key reagents. And if you want to connect with Lake Pharma, if you're an existing customer, please contact your account manager. That's actually the fastest way of getting a response and support. And if you're new to Lake Pharma, you can email either kitcovid@lakepharma.com or use the contacts page at lakepharma.com/contact us. And we definitely welcome feedback. We actually like to see your results if you're willing to share. And we're open to comments and suggestions, and you can give feedback. We have specific pages for that. And if you want to contact me directly, I would welcome that. And my email is hua.2@lakepharma.com. And that's the end of the presentation. We're going to go into a Q&A.
Thank you, Hua, for that presentation. While you look through the questions that have been submitted so far, let me just remind the audience, this webinar is recorded, and we will put the recorded version on our website very soon. So watch your email inbox, that's coming. And also there are a lot of other informations and tools on Lake Pharma's website, either for standard antibody services, antibody proteins, or plasmid DNA manufacturing. Make sure you check our website or reach out to Lake Pharma. Okay, so while I know we have some dozen or so questions being submitted, I'll let you go through the questions, and let's see how many we can answer right now.
Yeah, okay. So one question is, what is the titer with the production of the spike protein? So in our first webinar, we described that the various reports of the production of spike protein, the range is between 0.5 milligram per mil, 0.5 milligram per liter to 2 milligram per liter. It is a hard protein to make. In our tuna chill platform, we're getting in the neighborhood of 30 milligram per liter. And that is the reason why we're able to produce gram scale using transient. So ours is about 30. Second question, what tests were performed during stability testing? Well, that's we look at the SDS page, CE, as well as SEC a lot. From a functional point of view, right now we're mainly test using the ACE2FC binding. But as I mentioned in the presentation, we are coming up with a panel of antibodies, and we intend to use the antibodies as a way of testing the antigen as well. So the idea here is to validate the antigen, validate the antibody, so they can actually cross-check with one another from a product consistency point of view. The next question is, what protein domain is used to maintain the trimer structure of spike protein? Yeah. We actually think that the spike protein should, the reason it's a trimer is not because we make it a trimer, it's because the protein is a trimer anyway. When we look at the structure a while back, there's very, very extensive trimer interaction. The very extensive interaction of the spike protein, and from data, from experiment, we also see that the protein very much like to be a trimer, and we rarely see, I don't think we ever really see a monomer formation of the spike protein. So again, they're trimer because they like to be a trimer. This question is, does plasma work better than serum and whole blood? Well, I think that's probably, this is probably about the, well, we have only done work with plasma, and I'm not sure if this is about the serological assays or neutralization assays, but we primarily use plasma. But I believe serum would work as well. I'm not sure about whole blood. Next question, what differences did you see in ELISA reactivity to host spike and RBD? Yeah, this is actually a really interesting question. We're not seeing the same response or interaction between host spike and RBD. The profile is actually quite different. And when we test the antibody, we see even further differences. So it's not like RBD is just a simpler or cheaper version of a spike. They're different. We even see antibody that bind to RBD and bind to spike protein, but they're binding with a different kinetics and dynamics. So we're still examining this area. So what I can say is that from ACE2 protein binding, we can see the interaction between spike and ACE2 Fc. That binding is very, very strong. The binding between RBD and ACE2 Fc is much weaker. So now, is that a avidity artifact we're seeing? Well, we can't tell because we have no way of making a spike as a monomer anyway. And we have no plans to try to make a monomeric spike for comparison. So really can't tell. Next question is OC, not OC, but NL63 bind to ACE2. It's that we don't have the data yet. I don't know the answer. Next question, do you know if anyone has tested binding of spike protein in the non-ACE2 expressing cells? Does the spike protein get into cells do not express ACE2? I'm not the expert in that domain. I think weeks ago I've seen publications or news that there may be a second receptor for this, for the virus. Yeah, I don't know I don't know for sure. When we do the pseudovirus experiment, the target cells do express ACE2, and that's what I know. Next question, what is the expected false negative rate for ACE2 Fc ELISA neutralization assays? We're still early. We can't say for sure. False negative, I think it would be just my understanding of the assay and also the various experiments we have run, we do not expect to see a whole lot of false negative because this ELISA neutralization assay is so mechanistically clear. If it bind to RBD or bind to spike and prevent ACE2 from binding, I don't see how, well, we may be surprised to see false negative, but so far we have not. So that's from in that case. Now, this question may also say, could it be something that does not block the interaction between ACE2 Fc and spike, yet the antibody can neutralize, can work in the whole virus neutralization assay or the pseudovirus neutralization assay? Yes, then the answer is yes. You will see that because for sure there will be antibodies that can bind to spike but does not disrupt the ACE2 interaction, and they seem to have worked in the neutralization assays. So those are really interesting antibodies, and we look forward to analyze those antibodies in detail. The next question, what is pseudovirus? Are they virus-like particle? The answer is pseudoviruses are actually viruses. The pseudo is really not the best name for it. It's actually a virus, but it's a produced virus that usually is on a lenti platform and it's replication deficient. So it behaves like a virus. It can infect cells. You can have the spike protein on surface, but it does not. It's not capable of replicating. It's capable of delivering a reporter gene into cells, and that's it. That it cannot replicate. is pseudovirus neutralization assay from SARS-2 from patient previous diagnosed with PCR. Well, the pseudovirus assays are all man-made assays, and it's very popular in the virology lab. They make pseudovirus, and they look at the pseudovirus infection assay, and when you test things, it's neutralization assay. So those are not from patient. Will the slides be available? I know the video capture will be available. I'm not sure if this slide deck will be available. Next question is the SARS-CoV-2 protein is $450 per milligram. Is that for industry as well now? No, the answer is no. For diagnostic companies, for the commercial, for the industrial use, that's $900 per milligram. At $450 per milligram, we're not. We are. That's about our cost. So. In the previous, in the part 1 presentation, we actually ran the economics at this price point. Your ELISA assay reagent cost is less than 10 cents for the spike protein. We think the economics should work in our favor in that. Next question is ACE2 protein highly conserved across many species. There's actually literature on that, and I think somebody's probably making ACE2 protein from the zoo and sort of testing what can interact, but we're not doing that. I think that would be a scientifically relevant paper, relevant activity, but we're pretty busy doing what we're doing right now. We can't do that. There's been, but I'm sure the literature will come up on that very quickly. Next question, do we make SARS-2 pseudovirus for purchasing? If not, where do you get them? We are in process of making variety of pseudoviruses. They're not available for purchase. We generate our data via collaboration with Li Jinrong at the UIC. So they ran the experiment. Could there be pseudovirus for purchase in the next few weeks? Possibly. But we're not doing that. All right, the last 2 questions. Do you find furin cleavage affect the conformational stability of spike protein? I think that's a great question. I don't think anybody knows yet. And the reason for that is a lot of the majority of the studies that I have seen is using furin cleavage, using versions of protein that with the furin cleavage site removed. Engineered out in our case. So most studies done with that. And in order to look at the relevance of the furin cleavage, you really have to then compare to the wild type versus the engineered version. And we have those constructs, we have the protein. We're actually trying to address that using the pseudovirus assay. But right now that's still work in progress. Last question, is there any data to support the notion ACE2 is the sole receptor for SARS-2 entry? Put another way, how confident are you RBD blocking titers are representative of full neutralization titers? Well, right now I'm not confident, and the assays were done with the pseudovirus assay. That's still a bit, very well controlled in that we're not giving, if there is another receptor, we're not giving that receptor a chance. A good chance to show itself, so I can't say for sure. So the assays, most of the assays I show here is very much mechanistically, well, it's mainly on the ACE2 as a receptor. Could there be other receptors for this? I think they will. I think they will. But to say ACE2 is the main receptor, I'm pretty comfortable with that. To say ACE2 is the sole receptor for SARS-2 entry, I'm not sure about that. And if you look at the ADE part, where there's antibody-dependent enhancement, then you really can have a whole wide range of other receptors that could happen. So instead of relying on the virus direct binding to the ACE2 receptor, you can have the virus to bind somebody else, and that bridge a receptor interaction that really could happen. But for now, I think focusing on ACE2 is not a bad thing, because there's still a whole lot more we need to understand on the ACE2 side of things. And once we understand that, then we can look at what additional channels are there. So, all right, Walter, that's all the question I could see.
Right. Yep. I think we've got a few more from the chat channel. We'll have to take those offline. We'll follow up specifically on those additional questions, given that we're a few minutes over our webinar time already. So I want to say thank you to Hua, and also thank you to all our audience for this webinar. And we'll send an email notification as soon as the recording is up and available. Thank you again, and have a great day.
Okay, thanks everybody.